So this is going to be our first lecture as we start our discussion through these physiological principles of resistance training. As we build up to this discussion about some of the adaptations that occur and some of the important aspects to resistance training itself, we do need to go back and probably have a quick refresher on some of these structures and functions. So this is going to be some A&P review. There'll probably be a little bit of new material in here. But we're going to cover a lot. So we're going to cover things about the skeletal system. We'll cover the neuromuscular systems. We will then cover cardiovascular and respiratory systems. So there's a lot built into this. So it's going to take a little bit. So please be patient, but we'll get right through it. So when we start off this, if you can have an understanding of these four kind of ideas or topics, when we're done, you're going to be sitting very, very well for your first quiz and moving into your first exam. So understanding these structures of both muscle and bone, understanding the sliding filament theory, the differences in muscle fiber types, both from a morphological or kind of the size aspect and the physiological portions to that, and then having some idea about these anatomical and physiological characteristics of both cardiovascular and respiratory systems. So first things first, we're going to discuss the musculoskeletal system. So a mature human adult, right? As most of us are, we would be sitting with 206 bones. And the idea really behind this skeletal system is that we're providing leverage as well as support and protection. And when it comes, the support and protection is, I think, fairly easy to see and understand in terms of when you start thinking about things like your rib cage, your spine, right? The skull. Those are pretty straightforward in that they're providing support and they're giving you some level of protection. The providing leverage is where things can kind of get a little screwy. When we talk about movement, we're really talking more about the leverage aspect than the support and protection. So to kind of go into that, I guess, a little bit further, when we have a muscular action, that muscular action is not acting on an object directly, right? It's not, when you walk, you're not providing force from the muscle acting directly on the ground. Or if we pick up our phone, right, and we're holding our phone, the muscle's not directly acting on your phone. What's happening is that muscle's putting force into a bone, and it's pulling on that bone or providing some level of either pulling or pushing into the bone to then give us the movement that we were desiring or that we're intending to have. So that's an important part to kind of understand that goes into this leverage aspect. And we'll talk about levers a little bit more when we get into the biomechanical stuff on the next lecture. So if we look at the differences between our axial and appendicular skeleton, right? The axial skeleton is going to be anything that's really in the center. So we're talking about skull, vertebral column, ribs, sternum, right? That's in that center midline area. When we move outward to the shoulder girdle, so when we start talking about things like the scapula, we talk about sternoclavicular, the clavicle or your collarbone, moving out to the acromioclavicular or AC joint, right? That is, or your humerus and how that's acting in, or the glenohumeral joint at the shoulder, that's going to be appendicular. And then everything off of that. So the extremities are going to be appendicular, where everything else that's running kind of in the center would be part of the axial skeleton. So to take this into the next part, we've talked a little bit about bone. Now, when two bones are interacting with each other, or two or more bones are interacting, is a better way to put that, because that's what's happening in most cases. This is what we consider a joint. And so it's either a junction point or it's going to be a pivot point between these multiple bones. So a junction point you can kind of think of as your cranial sutures. It can be the SI joint, right, or your sacroiliac joint at the bottom of the sacrum coming into the pelvis. That's a junction kind of joint. There's not a lot of movement going on. We think about more of the pivot points, that this is going to be some of the things that you probably learned in either kines or you learned in A&P when we talk about our joint actions. Right? So when we talk about things like roll and spin and glide, when we talk about having different degrees of freedom and all that other good stuff, that's going to be the pivot point aspect. So movement of the body as a whole is going to be this rotation of these bones at an individual joint. So when we have whole body movement, so when you do something like walking, a very simple task, right? It's a rotation at the hip, the knee, and the ankle that's allowing that to happen, or allowing that transfer of muscular force to these joints and from segment to segment to interact with the ground to provide us our movement. That is the basis of human movement, right? Is that we have these rotations occurring at these joints, and these rotations are being generated by a muscular action. So that's how we move. Now, the job of a joint is not only to provide movement, right, or to provide this pivot point, it's also there to transfer or dissipate force. So we want, let's say when we walk, right, our bigger musculature in the lower extremity is going to be in the thigh, and we're going to have a lot of musculature on the anterior aspect of the thigh, or your quads. And those quad muscles provide knee extension. So when you walk and you have knee extension, we want that force that's being generated from that movement to be transferred down into the ground, right? So we need to transfer across the knee joint into the shank or your shin, right? Across into that, down through the ankle and out into the ground. But at the same time, when you do something like jump, right, and you go to land, you don't want that force to be transferred all the way up the chain and where you feel it, right, in your hips or your back or wherever the case may be. We want to have this dissipation of force. So that's an important aspect as well, is that in some instances we want to transfer force, in other instances we want to stop it from moving up the chain. So when you jump off your bed, or you jump off a fence or over a fence, wherever the case may be, right, we have this movement, but we also have these structures inside the joint that are going to dissipate the force as well. So we don't feel it all the way up into our hip and our back and into the spine. So that's an important aspect to a joint as well. So we can classify joints in one of two ways. They can either be classified by their structure, or they can be classified by their function. Those are the two classifications, structure and function. So when we talk about structure, this is going to be whether it's a fibrous joint, cartilaginous joint, or a synovial joint. Now, the function part is a synarthrosis or a diarthritic joint. So when we talk about structure, the first one function, we'll go into synarthrosis. And an example of a synarthritic joint is a fibrous joint, in that our cranial sutures have no movement, little or no movement. It's this, the fibrous, or these high concentrations of collagen going across the joint to hold everything together. Right? We don't have a ton of movement, if any movement, in the skull, or in the cranium itself, where these cranial plates are coming together. And this is really important, or very useful, in a transfer of forces. Right? So if you get hit in your forehead, that force doesn't only stay in that plate, but it actually gets transferred across the entire skull. That's an important mechanism when we start thinking about things like injuries and so on and so forth, that we have that. Another example of a synarthritic joint is going to be a cartilaginous joint. And cartilaginous joints are, they come in very, very various forms, but what it is this joint that is providing some level of movement, but it doesn't contain all the structures needed to be classified as diarthritic. These joints are typically going to be found in the midline, and they're going to have high levels of this fibrocartilage or this hyaline cartilage that is very movable and very flexible. And the two biggest spots that we can see this is going to be pubic symphysis, or where the anterior aspect of the pelvis where it comes together. So if we have the posterior pelvis being kind of that fibrous joint or that SI joint in the back, so let's hopefully see if this will work. Try to draw on here for you. So if this portion back here, right, if we're looking back there on both sides, if this is that fibrous joint that's being connected, right, what we want is that we need the anterior part to move. So we have this synarthritic fibrous or cartilaginous joint. Another spot where we see this is going to be the intervertebral disc going up the spine. So we're able to absorb force through there, but we also have high levels of flexibility with each one of those intervertebral discs, again, residing in the midline. So remember that we can classify things either by their structure or function. And so far we've talked only about the function aspect, and that would be our synarthritic and our diarthritic joints. So when we talk about structure, right, it's the fibrous joints and the cartilaginous joints both fall under the category of synarthrosis. Our diarthritic joints are going to be our synovial joints. This is something that I think gets mixed up and confused a lot, is people look at joint classifications and they'll see synarthritic and diarthritic. What the true classifications can be is either by its structure or by its function, right? So a diarthritic joint is extremely movable. There's no direct connective tissue link between articulating surfaces. So when we look at something like the knee, right, there's no direct connective tissue going between these. So we don't have that collagen that's crossing over the joint. We're also going to see that there's synovial, like a capsule or a cavity that's going to have synovial fluid in it. We have different axis of rotations. We can have lots of different things kind of moving on here, moving around with this. But the one thing that is common amongst all diarthritic joints, or to be classified as a diarthritic joint, is they have to have one of these 7 elements. So it needs to have synovial fluid, articular cartilage, a joint capsule, synovial membrane, ligaments, blood vessels, sensory nerves. You have to have all of those. If you are missing one of those, right, that means its structure is different, which means we're either going to fall into the classification of the fibrous or the cartilaginous joint, right? That's an important aspect, which then would give us a different function to that, which would be why we would have it classified as synarthrosis. So what we also have are secondary elements. So you can have things like fat pads, you can have a meniscus, you can have a bursa, you can have a labrum, right? All those are things that synovial joints may or may not have, right? But all synovial joints are going to have those 7 common themes of the fluid, the articular cartilage, capsule, membrane, ligaments, blood vessels, and nerves. So when we talk about this, these joints can move through a couple different ranges of motion. And it's really more than a couple, it's a few, right? So we can either have a uniaxial joint, right, or a joint that has 1 degree of angular freedom, and it only moves in one plane. So you can think of this as like a hinge joint. So a great example of this is your elbow, right? You can only have flexion and extension in your elbow. You can't have these other movements. When we talk about things like the wrist or the ankle, right, we have 2 movements at the 2 true movements at the wrist and the ankle that are in our planes of motion. So when we talk about planes of motion, remember we're talking about sagittal, frontal, transverse, those things. So you only have 2 angular degrees of freedom or 2 planes of motion that you're moving in. So right, so if your wrist, so if we're looking at this here, right, we can do flexion and extension, and then you can wave or ulnar and radial deviation. When you rotate your wrist, this is actually circumduction. So that circumduction is a multiple or multiplanar movement. So it's not classified as multi-axial because it's going through multiple planes. When we talk about multi-axial or a joint that has 3 degrees of freedom, right, and it can do all 6 of our general joint motions. So we can have flexion, extension, abduction, adduction, and internal and external rotation. And the 2 that we go to with that is the shoulder and the hip. Those are going to be the 2 that you would have those in. So again, this should be some review from A&P or Kines, and you should have a pretty good understanding of what just joint structure is kind of at this point. So as we move into the muscular system, one thing that is important to point out here is that we're gonna have lots of kind of layers of connective tissue in this. So each skeletal muscle is gonna have our contractile proteins, connective tissues, nerves, and blood vessels that are all a big part of what is going on with the physiology and the actions of the muscle itself. So again, this should be something that everyone should have at least heard once, twice, 3 or 4 times by this point, is that we're going to have 3 major layers of connective tissue inside of a muscle. So we have an epimysium, paramysium, and endomysium. So the epimysium is going to be around the outside or the entirety of the muscle. The paramysium is going to be around a group of fibers, right? And then when we come down to the individual fibers, it's the endomysium. So if we're looking at a figure like this, which is something you've probably seen 1,000 times in every textbook, in exercise science or athletic training or kinesiotherapy, right? Everyone has seen this picture at some point along the way. You can see that we have these layers. So our epimysium being at the top, take this highlighter off, right? So we have our epimysium here around the top or the outside, the endomysium around these individual bundles of fibers, or endomysium between the fibers, and the paramysium is going to be around the bundle of fibers. Excuse me. So just something that you look at. So moving from that tendon to the muscle belly all the way out to the myofilaments or our contractile proteins is what we're seeing here. So in addition to that, we're also going to have this cross-sectional view of a muscle where we can see that we have our myofibrils, but we also have these unique structures that make skeletal muscle. And that's the sarcoplasmic reticulum and these transverse tubules, or the T tubules. And those are going to be really, really important when it comes to making a muscle move, or to give us the movement that we're really looking for. So one of the things that we need to understand is that this is part of what makes skeletal muscle and different from smooth muscle and cardiac muscle. So this is important. But as we get to our muscle fiber itself, some of its big characteristics is that it's multinucleated, which means that a muscle fiber or a muscle cell itself is going to have more than one nucleus. And when we start talking about hypertrophy and adaptations, we'll get to that a little bit more in detail and why that's really important. It's also going to contain many, many mitochondria. Mitochondria being that powerhouse of the cell that we've all heard about over and over again. When we get to bioenergetics, we'll see why that's really important. But this transverse T tubule is a very special structure in terms of giving muscle fibers its defining characteristic. This is where we will see our excitation contraction coupling occur, and this release of calcium into the sarcolemma itself, or excuse me, into the sarcoplasm itself, which would then allow for muscle movement to occur. So we have our myofibrils, and then we have our intercellular structure. So anytime you see sarco, that is just telling you that it is the muscle aspect of the same thing we would have in other cells. So sarcolemma would be the membrane, sarcoplasm, same as the cytoplasm, sarcoplasmic reticulum, same as our smooth ER in a regular cell, or the cell as you've probably learned it through high school and out into biology in A&P. So myofibrils itself is what is going to give skeletal muscle and cardiac muscle its striated look and appearance. And what we are looking at is thick and thin filaments, specifically the actin and myosin. So actin is our thin filament and myosin is our thick filament. And you can see this arrangement that we have. So the dark band, when you look at an individual sarcomere, which is what we see here, and a sarcomere is going from Z disc to Z disc, is that darker band in the middle is going to be the myosin. And the actin is on the outside, and that actin is going to slide over the myosin. And this is how we get muscle shortening, and this is how we typically would be generating force through our sliding filament theory. We're going to get to a little bit more in depth into muscle movement and sliding filament theory here in a couple slides. But just having, again, this good, solid understanding what is going on at this point. This should, again, be a review for you. So if we're looking at this from a skeletal muscle perspective, right, we're seeing that we have our actin and myosin arrangement is not just a clear straight line arrangement, right? It's going to be three-dimensional. It's going to have this kind of circular pattern to it. So if we were to cut this at different points inside that sarcomere, right, so our M line, our I band, and our A band, as we have down here in this, you can see that it is this three-dimensional kind of patterning that goes around. So for every myosin, there's 6 actin that will act on that one myosin. Protein and this look that we have to it. And you can see in that M line that we have, this would be where we would have a high level of myosin. And that I band is where we're going to have the high level of where only the actin itself, and the A band is where that crossover is going to come. And when we talk about the M line, or that region itself, right, is in where that dark band and only the myosin, this is when we're at rest and there's no crossover between these filaments. But you can see what this would look like, right, as we kind of bring it down and look at these different structures. So what's really important now as we kind of move forward is we know what actin and myosin are. We know what is happening from a general structure of a muscle. Actin and myosin are sliding over each other to give us movement that's acting on our joints, which is then going up to the skeletal system, providing leverage and giving us the force output onto an object to have our movement. But from a physiological standpoint, how do we get there? And what we're going to go through now is our 14 steps to putting this together. So we haven't talked about the nervous system yet. We're going to talk about that next. But what we have is this action potential. And this action potential is going to be traveling down that motor neuron and gets to the muscle. That action potential, when it arrives at the nerve terminal, it is going to elicit a response where we see a depolarization. And we see these voltage-gated channels, specifically dealing with calcium, become important, and calcium is going to get dumped into the motor neuron or into this nerve terminal. And when calcium enters this nerve terminal, what this does is it triggers the release of the neurotransmitter acetylcholine. And acetylcholine is going to be kind of into these little buckets, I guess you would call them almost. And what it does is this vesicle is going to go all the way out to the edge, and acetylcholine is going to get dumped into the synaptic cleft or into this neuromuscular junction. And that acetylcholine goes across, and then it's going to attach itself to the nicotinic receptors on the muscle side, right? So at this point, all we know is that we've had muscle action potential, comes down the neuron, so our nervous system has said move, right? And that nervous system, by saying move, triggers this release or the increase of calcium into the motor neuron, which then dumps the acetylcholine across the synaptic cleft, right? So we can have acetylcholine then attached to the nicotinic receptors on the motor end plate. This acetylcholine gets recycled or gets broken down by acetylcholine esterase after it's had its attachment and binding with that receptor on the other side. So what that binding of the receptor on the other side does is this allows sodium into the muscle and potassium to leave. And what that flow of through our sodium potassium pump does is triggers a second action potential. So a lot of times we learn that there's just the one action potential coming down from the nervous system. But in reality, what it is the first action potential triggers the release of the transmitter to then generate the second action potential at the muscle itself. So you can have, in theory, right, a scenario where the nervous system is saying fire, okay, but if you don't have the right concentrations of sodium, potassium, and where they need to be, and that flow through that sodium potassium pump, we may have a blunted or a different action actually occur at the muscle. So there's two action potentials. That's important to remember, is that there's two action potentials. So now that we have our second action potential, what that's going to do is it's going to travel down the sarcolemma itself, or that membrane. And when it travels down that membrane, that action potential is actually going to slide into that T tubule that we talked about, or that transverse tubule, which is going to activate again a calcium channel. And this is where it's really important to understand that calcium is super important in terms of muscle physiology. When this calcium gets activated, or this channel gets activated, calcium gets released from the sarcoplasmic reticulum. So the T tubule is important because it's going to trigger the release of calcium out of the sarcoplasmic reticulum. That calcium that's now been released, or the second release of calcium, is going to bind itself to troponin, which will cause the tropomyosin that wraps around the actin to move or to change its shape. So troponin and tropomyosin are they do wrap themselves around the actin protein itself. And so calcium comes in, has that attachment, and then to troponin, which then moves it. And so tropomyosin is there. And this reveals the binding site. So this is when we have this movement or this change in that structure, this is how myosin and actin can now come together. So to have our cross-bridging, or for muscle movement to occur, this calcium's got to be there. So the last steps that we have here, right, is that now that this change has happened, as you can see kind of the top of this figure here, this is at rest. Calcium's now been bound to the troponin, which then moves our tropomyosin and opens up that binding site. And at this point, the myosin head is going to bind in and attach to that actin binding site. And on that myosin head you have the inorganic phosphate and our ADP. And that ADP and inorganic phosphate is important at this point, because when we dump the inorganic phosphate, or we release it off that myosin head, we get what we call the power stroke, where you actually get the movement of the actin itself. And so that myosin is basically going to attach itself, dump the phosphate, and then you get movement. So what ends up happening at this point is the phosphate and ATP come back together, make ATP, which when that binds to that myosin head, right, what it does is it creates a release. So this actin and myosin is going to kind of work as a trigger system, in that the ATP binding in is going to release the myosin head from the actin. So if you don't have ATP come back in, what ends up happening is that binding stays in there, or it doesn't trigger a release. So when we talk about things like rigor mortis, when someone dies, right, and their muscles tighten up, this is what happens, is that you're going to have all this calcium get flooded into the muscle, and then you have that change in the actin itself. So the ADP and that phosphate come in and they attach, and you can have some movement there for a while because of our energy systems still breaking down glucose, still breaking down some fats, your creatine phosphate that's in the muscle, right? We have all these energy systems that are there that all of a sudden what ends up happening is we no longer can produce ATP to allow that binding to be broken apart, right? So as long as ATP is there, it breaks apart the actin and myosin. Then when you break the ATP and you get that release of energy again, right, then we can have the head attach. And this is going to continue and cycle until calcium is no longer present. So as long as calcium is present inside the muscle, you will have this cycling occur, right, where you're using ATP and you're using energy to make this muscular action occur. When the action potentials have stopped, right, and they've stopped coming from the nervous system and they've stopped coming from that second action potential along the sarcolemma and entering the T-tubule and into the sarcoplasmic reticulum, and we no longer are triggering that release of calcium, what we have is the calcium ATPase pump, which is going to bring the calcium back out of the sarcoplasm and into the sarcoplasmic reticulum, and this will end the cross-bridging cycle. So I know that's a lot there, right, in those 14 steps. But those 14 steps are going to be really, really important for your first exam. You will have a question on those 14 steps in terms of listing what is going on. 1 through 14, I want you to be able to do that. You've got plenty of time to study for that. Our first exam isn't for at least a month. So plenty of time to kind of put that in and study that and know what's going on there. So number of cross bridges that are formed between actin and myosin is going to instantly tell us, or at a specific instant is going to dictate force generation, or how much force is being produced. The more cross bridges you have, the more force that you have. So at the early stages of muscular actions, there's not a ton of cross bridging, right? Or when we're at elongated or at the full range of motion. So if you can think of, I know this is kind of bad, but sitting here like this, right, full range of motion, that bicep has now been stretched out. So there's not a ton of cross bridges there. But as we get closer and closer and closer to bring the forearm up, right, and now we're starting to have this alignment, now I can produce more force, right? And until I reach a certain point where now that actin is actually going to be too far and it starts sliding across, and now you have too much overlap. That goes into our length tension curves, and we'll talk a little bit about length tension in the biomechanics lecture. But have this understanding of the actin and myosin, how they cross over each other is going to dictate force. So calcium and ATP are going to be critical and are necessary for this cross-bridge cycling to occur. So without those two things, you cannot have muscular action. So now we move into the nervous system a little bit, and we're going to talk at first about the motor unit itself. So a motor unit is a alpha motor neuron, and it has its origins somewhere in either for the most part, for what we're going to talk about, and to simplify it, it's going to be starting in the spinal cord or in our central nervous system, and then it reaches out to the muscle. And it's going to innervate a large number of fibers potentially. Right? So a motor unit is the motor neuron and all the fibers it innervates inside that muscle. So that is what the kind of the classic definition of a motor unit would be. So it's important because remember, this is where that action potential is being started. So the central nervous system itself is going to give us the control over those muscular actions that we have, and to start that whole process that we just talked about in those 14 steps. It's going to be triggered by this activation of a motor unit. So this is a little bit of a kind of nerdy slide, I guess. I think they're kind of cool, this slide and the next one, but this is actually showing you the alpha motor neuron, how it's stretching across some muscle fibers and what this would actually look like in terms of our neuromuscular junction. So when the alpha motor neuron is joining in and kind of wrapping itself over some fibers, and then where that connection is our neuromuscular junction, which is where that release of the acetylcholine is occurring. Same thing is going on here. You can see that kind of the bundle and how that is interacting. So what we have next in terms of a discussion and to understand what is the function and some of the physiology behind these structures is this discussion about action potentials itself. So when an action potential occurs, it is that signal coming down from the nervous system. So what we are doing is we're changing the polarization of these fibers or of these, the nerve fibers. And when we hit a certain threshold or a change of 15 millivolts in that threshold, this is what activates or generates that potential to move all the way down the motor neuron itself. So you have to have this change in potential to trigger this system or trigger these steps. This is typically an all or none event, which means that if that voltage threshold gets hit or has been crossed, then the action potential is going to occur. We see the changes or those steps occur. If you have a signal but it's not strong enough, right, or that input isn't strong enough into the nerve itself, we don't trigger the action potential. So this is what your typical action potential would look like, right? So we're sitting at rest at about negative 70 millivolts, and then we have a stimulus that's applied. So this is coming from a dendrite from another neuron somewhere inside that central nervous system. So if you can kind of think about this, right, you're going to have a connection from the brain and the motor cortex of the brain all the way down the spinal cord, and eventually it's going to hit a motor neuron to whatever muscle we're talking about. So when that signal gets sent from the higher brain centers, right, in that motor cortex and goes down the spinal cord, and we get to this point and it says go. If that stimulus is large enough to change this potential by 15 millivolts, then we see this huge shift, right? So we go from this negative 70 all the way up to a positive 30. So a big change in the polarization of the nerve itself. And this is the action potential that we're talking about in terms of what's traveling down the motor neuron and getting to that terminal junction or that terminal motor or the nerve terminal, that is triggering that the first influx of calcium and then the release of acetylcholine, right? And when that voltage falls, so it's going to go up, it's going to trigger, and then it's going to come flying back down. What it actually ends up doing is it goes below our negative 70 or falls below that resting potential as some of these polarization or these charges get shifted, right? And then we get back to our resting spot until it goes again and occurs again. So when we talk about this as an all or none principle or all or none mechanism, what this is again, you can see that there's some sort of stimulus here to that nerve. It just wasn't big enough to trigger a release. If it's good enough or it's a change that is large enough, right, you're going to get this full potential. That's what we see here through the good enough, right? And we see it go down, and then we're back at rest, and we're hanging on. Now we have a super large stimulus, right, coming, and what ends up happening or occurring is it still gets to that threshold. So you get the same response, whether it barely gets to the threshold, right, or if it's way over the threshold. That nerve has the same response to it. So this is where it's in everything that the nerve has, or it's nothing at all. And it's going back and forth. So how we evaluate a twitch or an action potential is in one of three ways. We look at this through the force generation and the time response to a single potential. And what we call this single potential is a twitch. So when we talk about twitch interpolation, this is to give us an understanding of the dynamics of this motor unit. So you would have potentially a very specific alpha motor neuron characteristics that would go to specific muscle fiber types. And that's really important to, I guess, see, right? So the way that we do this is through the time to the peak force, the amplitude of that peak force, and the half relaxation time, or your ability to get back to baseline. So when we look at a twitch itself, right, we're going to have a couple of things that we can point out on this slide. So the first is that you have a stimulus. And that stimulus, when it occurs, it takes a period of time to start having this ramp up from force production, right? And so that's called the latency period. So when we talk about things like a fast twitch muscle, the latency period is going to be very small. It's fast. When we talk about a slow twitch muscle, well, slow twitch muscle just means that it takes longer to get through that latency period and through the contraction period. So this part here is that time to peak force, or that first identification of what type of motor unit are we dealing with, right? Second, off of that, is we can then look at how much force or how much tension did that twitch or that action potential generate. So if you have a muscle fiber, or you're looking at two motor units, we'll talk about it through this way. If you have two motor units, you have motor unit 1, motor unit 2. Motor unit 1 and 2 have almost identical latency contraction period timestamps, or that time to peak force. But motor unit 2 produces 300 extra newtons of force. That additional 300 extra newtons of force now can separate out a type 2A versus a type 2X muscle fiber. Right? So again, this is how we can start determining which motor units are associated with which fiber types. So this is where that's important. So we have our time to peak force, the actual peak force, and then what's the relaxation period? Or how long does it take to get back to half of that force generation? This, again, has to do with our fiber typing, in that a slow twitch fiber is going to be able to send that signal again quickly, right? We're getting it faster and faster and faster. Or a fast twitch fiber, you get this huge force output, but it's going to take a little bit longer to get to that half relaxation time and to be able to trigger that again. So when we look at this from a motor unit force generation, it's how do we put these action potentials together? So we know that a motor unit in the nervous system just doesn't send one potential, right? When we talk about understanding motor unit types and muscle fiber types that are associated with that, we can do that by sticking an electrode inside there, attaching it to the muscle fiber itself, and then us control it and send the signal. That's how we can interpolate one action potential. But what in reality happens inside of yourself is that this action potential is sent over and over and over again when we really are looking for muscle movements, right, or joint movements. And these overlapping twitches, what they end up doing is building off each other into what we call tetanus. So when we look at tetanus, right, you have one potential. So this is your force generation out of that one potential. When you have two potentials sent, right, what we end up seeing is what we call a wave summation. So this potential generates this first increase in force. As it goes back down, or starts to go back down, the second action potential has been sent. So then it summates or adds to it. We then would come back down. Unfused tetanus is when we kind of have that a twitch in a muscle, and that like if you were sitting and your muscle twitched, right, and it would just kind of go back and forth, and you'd have this muscle twitch, right? in that you'd see this unfused kind of hump pattern to it, right? In that it's going to build up, but then goes down, builds up, goes back down, builds up, goes back down. And what we really would like to see is a fused tetanus, where this is all coming together into one force generation. So if we were to look at maximal force generation out of a motor unit, right, and you have this action potential constantly sent, it's going to eventually plateau in that that muscle unit or motor unit and muscle fiber can no longer produce any more force based on those action potentials being sent. They're going to keep adding together. We get this nice straight line of force generation, but then it plateaus. So again, our ability to identify, is this a fast twitch? Is this a slow twitch? And that peak force potential that we would have. So this is the end of our first section. We'll dive into some more of the motor stuff and dealing with motor neurons, and then we'll get into our cardiovascular stuff in the second lecture.